Good morning, good afternoon, or maybe good evening. Welcome to this session on advanced techniques for functional and structural neuroimaging of the cerebellum. It's a special session at HBM. We see a lot of cerebral imaging, but the cerebellar stuff tends to be a little bit less highlighted. So we're very pleased to present in this session. So what is it, the cerebellum? It's a little brain, literally. It's essential for a lot of our daily tasks, motor control, motor learning, but also for more cognitive things like speech and emotion control even. And the main problem of cerebellar imaging is that it's usually not, or maybe only partially included in neuroimaging, where we tend to sort of stick to this red box that you see here. And effectively, that means that we're not looking at the entire brain. It means that we're looking at something more like this. And even if the cerebellum is included, then it's usually not in a sufficient resolution to look at the gray matter layer. And that's where all the action happens. So this is a shame. And if we want to really understand what the brain is doing, then we should look at all of it, including the little brain. So in today's session, we're going to share some of our tips and tricks with us and show you that it's possible and not even maybe as difficult as you thought to acquire good cerebellar data and analyze it properly. I'm going to start off with talking about high resolution, high SNR in the little brain, why that's important. Then Henrik Lundell is going to take over with a talk on diffusion imaging and why that is especially difficult, as you see here in the cerebellum, and then also on how to actually make good on that and acquire data as beautiful as you would like. The third talk is again diffusion, but then tractography rather than just diffusion imaging, and looking at the pathways that enter the cerebellum and how these change as here a function of the heartbeat. In the second half of the session, we're going to look at data analysis. Joern Diedrichsen is going to kick off with the suit toolbox. So he's going to talk about functional maps and functional segmentation of the cerebellum and the atlas that exists for you to reference your own results to. So you see a nice example of here, the flattened out map. Then Pierre Bazin is going to talk about cerebellar segmentation, so really separating that gray and white matter, even though it's so close together, and generation of cortical maps, inflated maps like you might know from the cerebral cortex. And the last of the presentations given by Anila DeMello, who's going to talk about the role of cerebellum in language and neurodevelopmental disorders. And this is more of an applied example of the importance of the cerebellum in a task that you might not know the cerebellum was involved in, but that it's involved in very different ways and very extensively so. So with that, I'd like to hand over to the speakers, and I hope you enjoy the session. Hello, everyone. Good morning, good afternoon, maybe good evening. It's my presentation on high resolution and high SNR in the little brain, also known as the cerebellum. I'd like to start with pointing out why that high resolution and high SNR is so important. So this is a single slice through the cerebellum. Down here, where you can see that the cortical surface of the cerebellum is very, very thin, especially compared to the visual cortex here above. Now, the visual cortex is already not a region that's known for a very thick cortical layer. It's only about 2 millimeters here, but the cerebellar cortex is only about 0.5 millimeter. So you can imagine that you need a very high spatial resolution to properly see that, and most of our standard methods are not really up to this. So we need to focus to actually bring that resolution, to bring that cortex sharp into view. Another feature of the cerebellar cortex is that it's very, very folded. So where this cortex is relatively flat, and there are stretches where it'll just go on, there's a lot of very tightly packed wounds here in the cerebellar cortex. One of the advantages of looking at the cerebellum is that if you do manage to visualize the cortical gray matter, that you can also see the layers inside that cortical gray layer, which is much more difficult in the cerebral cortex. So why is that such a problem, this resolution? Well, MRI is an inherently low SNR method. The signal that we're working with is small, and in humans, it's even smaller because we have to work at room temperature. So what you also have to remember is that voxels are cubic. So going only, say, half from 1 millimeter to half a millimeter is kind of a large step in terms of voxel volume. Now, if we're comparing this 1.6 millimeter image where there's a lot of signal with very little definition, you can't even really see that there is a cerebellum in there, to one that's taken at 0.8 millimeter, and we see a sharpening, we see more details, we can see some of the white matter bundles stretching here throughout. It's sort of becoming a tree-like shape as we know it, but we certainly cannot segment the gray from the white matter. If we go to a voxel size of 0.4 millimeter, which is already much higher spatial resolution than we're used to, then we start to see that gray matter layer more properly, but it's still not quite sufficient. Now we can take one more step up in spatial resolution without our volunteers complaining too much, and then we get to a 0.2 millimeter voxel isotropic. And now we might be able to see the gray matter layer everywhere if we would have enough signal to noise. There are big patches here and around here, when even down here, something very strange is happening, where we simply don't have the signal power anymore to distinguish the tissue types. So it's a very thin line between having enough signal and then having enough spatial definition to be able to see what's going on. What you can also see is that this 0.2 is maybe not quite enough. Another problem is that high resolution means that we need to acquire lots and lots and lots of points in K-space, and this takes time and then we're not talking about minutes, but maybe an hour or maybe 2. And if we go even higher spatial resolutions, that gets even more difficult. So if we want to get a ideal cerebellar image, then we really have to think of getting more signal as well as more spatial resolution. Now, of course, it is possible to acquire very beautiful, even 100 micron isotropic data, which is a figure that I got from Joern Diedrichsen as an example. Now, unfortunately, it took, I believe, more than 24 hours to actually acquire this data. And of course, in vivo, that's not possible. This is an ex vivo sample. So there is a way, but it's very, very slow. Another way of getting more signal in an MR image is by cooling. And this, again, cooling your sample may be possible, but probably not very ethical and certainly not practical. Then that leaves the third only option, which is to use a stronger magnetic field. So given that the other two options sort of fall off because they're in practice too difficult, I opted for stronger magnets as the subject of this talk. And with that, I mean, especially the ultra-high field scanner. So this is from 7 Tesla and up. And in my lab, we have a 70. So the examples I'll be showing are 70 images. And there are, besides the main field strength, there are a lot of MR properties that change as you move up in field. There's first, and maybe most importantly, the higher SNR, which goes up with a factor a little bit above 1. And also importantly, the increased susceptibility contrast and the increased to stark contrast. Now, if you're interested in functional imaging, as I am, then those are two very important properties. There's also a feature or a challenge, if you like, of the B1 inhomogeneity. And because that's really important in the cerebellum especially, I also want to pay some attention to this. So if we're looking at high field and high SNR, then, well, high SNR, how much higher does the SNR really get? And I borrowed this nice figure from a paper from Pullman et al. In 2016, where they scanned people on the 3T, a 7, and a 9.4. And to save space, I just showed the 3T and the 7T images here. And if you look at them just by looking at them, you can already see a lot of differences between these two, even though it's the same brain. There's obviously a certain grainy quality to the 3T, which is not there in the 7T. It's like the signal jumps out stronger at you. There's also a little bit more contrast between the gray and the white matter in the 7T than there is at the 3T for this particular acquisition. It's T1 weighted, I believe. And there's this gross sort of shading over the brain. There's a dark shadow here and reaching up here. And these are the B1 inhomogeneities, or the effects of the B1 inhomogeneities that I mentioned earlier. So going back to the SNR, even with these shadows, we see that there's a massive increase in SNR everywhere. So if we look at the different brain regions, cerebellum's the green one, then here, then it goes from, so after I remember, around 600 to maybe 2,400. So that's a factor 4. That's enormous in terms of how much longer you would have to scan. This is really a big difference. So the amount of signal that you can acquire in the same scan time is then massive. Now, in addition, like I said, if you're interested in functional MRI, as I am, there is also an increase in the bold signal. So this is an old figure paper from myself, where we looked at a single subject who we scanned at 1.5, 3T, and 7T, and they were doing a very simple motor task. So this is a single subject, 1 ROI, the same bit of tissue. We extracted a time course, roughly the optimal echo time per field strength. And then you see that between 1.5, 3, 7, there's almost a linear and even a bit larger than linear increase in the bold signal. So not only do you have more signal, but you're also you're using that higher SNR to look at a signal which is bigger. Now, again, this is a primary motor cortex. So does that work in the cerebellum as well? And luckily for us, yes, it does. So we see much stronger clusters in the cerebellum than we do at 7T than at 1.5T. And that means that we can scan for shorter. We can also trade the time for doing more tasks in the same session, or you can go to higher spatial resolutions, which, as we saw before, is always convenient when you're looking at cerebellar data. Now, the shadow of lying over the cerebellum is the B1 inhomogeneity. And you can see it here in this image, depending a bit on your screen settings. It might look like a black hole, which is sort of wiping out, especially the lower, the posterior lobe down here. But it's clear that there is less signal in this stroke than there's here. It's more pronounced on the right than on the left, but it's always present. So one of the ways that you can get around or get rid of this shadow is by focusing exclusively on the cerebellum. You can do that, for example, by taking a local receiver. So we have an example here from 2015. Bazin, the little paper in Utrecht, they took a bunch of very small local coils that you can position in the back of the head, either under the visual cortex or at the cerebellum. The advantage of using such small local coils is that because they are small, they can be very close to the head, and that way they pick up a lot less noise because they're not sensitive to the area, the empty space inside the coil. But again, being local, it means that their sensitive area is limited. You can see the cerebellum, maybe the brainstem, but then not a whole lot more. And this is, of course, not always convenient. So here's an image that shows just how local that signal advantage is. We see a big increase up to about like 3 centimeters from the skull, so roughly to where you would find the hand movement region, for example. So here in green, so these regions all benefit from having local coils. If you move further forward, then it's maybe more difficult. So we looked at one of these ourselves, where we're looking at a cerebellar coil where also the transmit was local. And then you see that there's an even bit more stronger increase towards the surface, which then also translate in a functional advantage or functional signal boost in these 2 participants. Now, again, local might not be all you want. For example, if you're studying the motor system, then you might be interested in the cerebellum, but you're probably also interested in the primary motor regions. So then having a local coil is not the best solution, and you're better off probably looking at extending the sensitive area of an array coil, a head coil. And you can do that by using a dielectric pad. So these dielectric pads are, well, they look a little bit like a sandbag, but they're filled with a dielectric material, which sort of pulls the B1 a little bit towards the area where you place it. So if we're again, focusing on the cerebellum, then we place it at the base of the neck. And then we see that there's a signal increase close to the coil. There's probably an associated signal drop in the areas which have a lot of signal to start with. So here, but these JMRI paper here showed nicely that there's more signal here, and that this is not accompanied by an increase in SAR. So we're not changing the safety limits of the coil in any way by adding these. So we've also done these ourselves. We look here at a T1 weighted image where we see a gray shadow over cerebellum through the temporal lobes. And then we see that it's also really passing the area where you would expect a functional response to a finger tapping task. Now, if we add the pad, so this is a very small intervention, it's just a different cushion in the neck of the volunteer, then we see that there is now suddenly a cluster in the posterior lobe, which was not there before. Still, it's better, but it's maybe not fixed. So we have more options, luckily. We can also use our knowledge of the MR system, and instead of using a single transmit system, look at multiple transmits. Now, these are a bit like your surround system compared to a normal stereo. If you place more speakers around your chair or more transmit coils around your head, then you can control better where the sound, or in this case, the B1 goes. So we can look at the interference pattern that we have, and then we can change the phases of those B1 pulses in such a way that they no longer interfere as badly. So just because it doesn't really look like a music system, this is a schematic of a head coil, where an 8-channel coil with 8 loops positioned around the head. As you can see already, these are say you have degrees of freedom in the left-right axis and in the anterior-posterior axis, but in the Z-axis there's only one coil. Right. So if we now change the relative phases of those elements, we can tease away that black hole a little bit, and then we see that the head coil, when it's shimmed, performs better in terms of bolt signal, but also in terms of B1 mapping or temporal SNR or whatever measure you throw at it. But in this case, the bolt is maybe the most important one, so we see that it's more balanced. Right? These clusters in the left and right are more equal. Now, like I said, there's no control in this way on the Z-axis, but we do also have a gradient system that allows us to tie into that pattern. So if we think of a transmit K-space, and that sounds maybe a bit vague, but you're all used to acquiring images in K-space. We know how that works. So we're looking at spatial frequencies. And if we're acquiring spatial frequencies, our center dictates the contrast and the brightness of the image. And as you move out, you look at higher and higher spatial frequencies. So if you think of a transmit K-space as a way of imposing the same control, but on the spatial pattern of the excited slice or slab, then moving your K-space, your excited point in K-space away from the center means exactly that. So now we don't have a homogeneously excited phantom. There's a ball phantom behind here, but we have superimposed on the shape of the ball, we have a sinusoidal pattern. So it's insensitive to the B1 field in a shape which is dictated by the position of our excitation point. Now you can imagine that if you do multiple points, you can combine these, much as you use multiple points in K-space to combine your images, and you can form a pattern which fits the B1 pattern that we know. So if we look again at our image with the unfortunate dark area around here, then we can say, well, here is where we'd like to have more signal, and we excite this area stronger to achieve a homogeneous flip angle throughout the brain. So I'll just flip a few times between these two. So if we're acquiring data in a standard way, then we see a signal loss. If we're then using this KT points pulse, where we're exciting points in the transmit K-space, then we see that there's not only in the anatomy more signal, but there's also now a much stronger activation cluster in the cerebellum. So just to do that on a little bit bigger image, we're comparing the same brain, same region within that blue line. There's not only more signal, but it also translates into more bold signal. Okay. So one important thing to save time is to do your experiments cleverly. If we're thinking of high resolution, then it's slow, just because you need to acquire so many points, and it's also very motion sensitive. Now, one of the ways that we are dealing with this is by combining T1 and T2 measurements. We started from the MB2H sequence, which is a standard ultra-high field acquisition method, generating T1 weighted images and T1 maps. And it's relatively insensitive to DB1 unhomogeneities already. And we added to that this multi-echo gradient echo block, extended the second readout to be multi-echo, to have a T1 and a T2 star measurement in one. Now, so we acquire this whole bunch of different images. We obtain the T1 from these four, a T2 star map from those, and the finally QSM from the phase of the multi-echo data. And having all these together means that we can segment well the gray and the white matter from the T1, but we can also segment the deep gray matter structures and visualize the dentate nucleus, for example, in cerebellum. What we also have, because we have a little time over, so it's efficient because we're acquiring two at the same time, but there's still a little gap, and we added in that gap a fat navigator, which is an EPI image, only exciting the fat. And with that, and we get a full volume every TR. So these are lines, or maybe plane in K-space, and then here we have a full volume. And that means that we have a time course. So these are fat navs, and then this is a time course. So you can actually see how the subject is moving over time. And this being EPI data, you can feed it to SPM, and then it returns motion parameters, much like it does for a normal fMRI data set. If you look at these, and you have for 6 degrees of freedom, you have a very good estimation. And with that information, you can then go back to the raw data of the slowly acquired anatomical scan and put the K-space points back in the place they should have been had the subject not moved. You can also do this in a prospective manner. There's very nice work on measuring the motion as you go and then updating the gradient system. We chose to do retrospectively. Nice thing of doing it retrospectively is that you can compare with and without motion correction. So here on the left, we have a subject, which it's a T1 weighted data set that looks pretty usual, not very alarming, until you look at the right, which is the corrected image. And then you see that there are large areas which are actually quite different. There's a fuzzy boundary here, which is really sharpened up. Most of the gray white matter boundary is actually sharper on the right than it is on the left, but also the vessels are especially noteworthy. So there's a vessel here, which isn't even visible without motion correction, but also the vessels that are there, so here and here, they suddenly become more vessels. I mean, we know that vessels are round. They should be round if they cross your plane, your imaging plane. They should not be fuzzy stripes. So this gives you a grip on motion correction. It also then allows you to scan for a bit longer, because people will stay still, or it doesn't matter so much if they don't properly stay still. Also for subjects that move more, and this is of course a nice example, also including the cerebellum. This is before correction and after, and you really see the sharpening up also in the pons, but especially that cerebellar white matter tree is a lot more focused with the motion correction. So just to summarize all that, high resolution and high signal are very important to properly bring the little brain into focus and to achieve an imaging coverage like this. Simply extending your slices is a very, very good step to start with, but maybe it's not quite enough. Having ultra high field helps you because you get more SNR, and that pays off in getting that higher spatial resolution. You can achieve very long averaging times ex vivo, of course, but also with motion correction. maybe giving you the room to acquire from the same subject for longer to still have enough SNR to bring your small anatomical details properly into view. And the inhomogeneities in the achieved flip angles, which are always present at the ultra-high field, can be dealt with using various strategies such as dielectric pads, which are simply added to an existing RF coil, parallel transmit solutions, or using local coils. So with that, I hope I've given you enough ways and pointers of getting your own beautiful images from this nice region of the brain. I should acknowledge all these people for borrowing slides or ideas from them. And then I would like to thank you for your attention.
Hi, I'm Christopher Steele, and I'm going to tell you hopefully some very interesting information about the structural connectivity of the cerebellum. So my lab is at Concordia University in the Department of Psychology. I'm the Neural Architecture, Behavior, and Connectivity Lab. And some of the work that I'm going to be presenting, some of my own work that I'm going to be presenting towards the end of this comes from a work that I did while a postdoc at the Department of Neurology at the Max Planck in Leipzig. So with that, I think the first question that everybody should be asking in this type of a session, since it's an educational session, and thank you for attending, is why structural connectivity? Why do we care about structural connectivity? We've got that fMRI thing. We can do resting state functional connectivity. Why would we care about structural connectivity? Well, I think the most important argument for why we should care about structural connectivity is from the fact that the function of a region is predominantly determined by its local structure and its connectivity, right? So the inputs to a region determine what happens, so you know the type of information that's coming in, and there is computation that occurs within that region, and then the output is sent away, and wherever that output goes, obviously it's directed at a particular region where that information will be useful, or should be useful. So I think this is a really nice illustration of it from Bazin in the early 2000s, and these are what are called connectional fingerprints. And we have two different areas here. We have area 14 and area 9, and these are in non-human primates. So this was invasive tract tracing. This is over multiple animals. And here what you can see is that those two regions have differential connectivity. So going around the outside of that plot are different regions of the cortex. And we know that area 14 has very high connectivity to 10, a little bit less to 11, and almost none to 8B, for instance. And then in area 9, we have afferents that are connecting more to 8B, and obviously different if you go all the way around. So these connectivity fingerprints give you an idea of the function of the region, and the connectivity, so the inputs and outputs to the region, are determining what the function is going to be. So now we know why we care about connectivity. Why do we care about connectivity in the cerebellum, other than what I just told you? So I think the cerebellum is a really interesting structure because it accounts for over 50% of the neurons in the brain, and it supports the behavior, our skilled behavior, through its reciprocal connections to the cortex. So it's highly interconnected with the cortex, and it's highly interconnected not just between, not just to motor regions, but to non-motor regions as well, such as language regions and other regions for cognition. And this is something that Jeremy Schmann's group worked on for many, many years, and he really started to change the field in thinking about the cerebellum and its non-motor functions. I think there's another really interesting talk in this educational section where you're going to see some more of that. So we have these connections to motor and non-motor regions. And another interesting fact about the cerebellum is that it has a very uniform cortical structure. And because so this is across the layers of the cortex, because this cortical structure is so uniform, connectivity is the key. So the regions themselves are going to be doing the same type of computation. It's just the inputs and outputs that's going to make a difference for the functioning of the region. So that is really, really important. Connectivity is the key for the cerebellum in particular. So this map that I've shown you over here on the right is from a review article by Narinder. And here in blue, we have regions of the cerebellar cortex that are thought to be predominantly connected to motor regions, sensory motor regions. So in blue, we have the superior portion of that, and then flipped representation of this in blue down here in the lower part of the cerebellum. This is a sagittal view. And then we have Crus 1 and Crus 2, and these are commonly thought of as non-motor regions of the cerebellum. And I'll be referring to these motor and non-motor regions a little bit throughout the talk. But also at the end of the talk, which I'm going to sort of preview here, and I think you're going to see a lot of in a couple of other talks within the session, is that this distinction, so these are called lobules, the lobules of the cerebellum, this distinction between motor and non-motor lobules of the cerebellum is largely artifactual, and we should be thinking about it in more nuanced terms, which is what we should be doing going forward for connectivity as well. But having said that, most of the work that I've done now has been based on this lobular type of approach. So an overview of cerebellar connectivity is actually relatively easy because we have one primary output towards the cortex, and that is the superior cerebellar peduncle, and that's down here. So this would be the this major tract, or this peduncle, arises from the deep cerebellar nuclei, predominantly the dentate nucleus, and that projects up and through to the thalamus, and then from the thalamus to the cortex. Otherwise, we have, so the primary is directly to the thalamus. So inputs to the cerebellum come via the cortex. So the cortex projects down to the pontine nuclei. The pontine nuclei then project contralaterally through to the cerebellar cortex and to the nuclei. So we have some projections going to the nuclei and some continuing through to the cerebellar cortex. And this is the primary route of information coming from the cerebral cortex to the cerebellum. We have peripheral input up from the brainstem and spinal cord coming up through the inferior cerebellar peduncle. And that's here in blue, coming up like this and terminating throughout the cerebellar cortex as well. So again, we have the output from the superior cerebellar peduncle. We have the primary input from the cortex, from the that comes via the pons along the middle cerebellar peduncle, and we have peripheral input that comes up through the inferior cerebellar peduncle. So I'm not going to talk very much at all about the ICP in this talk. I'm more brain-centric, so we're going to focus a little bit more about the cortical and the pontine sides of things. All right, so that's the general overview of the inputs and outputs along the peduncles to and from the cerebellum. But as I mentioned before, so here's another depiction from Neuroimaging showing this reciprocal connectivity between the cortex and the cerebellum. And here we should have the little nucleus, or the deep cerebellar nuclei, included in here, because the cortex, cerebellar cortex, is projecting to the cerebellar nuclei. The dentate nucleus is projecting up towards the thalamus, and then the thalamus on to the cortex. And then the cortex is projecting back to the pontine nuclei, and from the pontine nuclei back to the cortex of the cerebellum. So those loops occur reciprocally. So one region, sort of this motor region of the cerebellum, will be projecting to a motor region of the dentate nucleus, which is going to be projecting to a motor region of the cortex, and then eventually back from that region of the cortex is projecting back to the same region of the cerebellar cortex. This is what we mean by reciprocal loops. Okay, so that's kind of the overview, and you can really zoom in on that and look at the cerebellar microcircuit, which is what I've shown here. So I don't want to go into too much detail here. This information is very well known, but just to highlight here that the inferior olive is the primary input along the inferior cerebellar peduncle, and this gives rise to the climene fiber inputs to the Purkinje cells. And the Purkinje cells are the major output, or the only output, of the cerebellar cortex. The MCP comes in, the information comes in along mossy fibers. They synapse with granular cells in the cortex, again, give rise to the parallel fibers that modulate Purkinje cell outputs. The MCP also, as we said before, has projections that go directly to the deep cerebellar nuclei to modify activity here. And then the output from the Purkinje cells synapsing on the cerebellar nuclei is predominantly through the SCP up to the cortex, through the thalamus. Okay, so that information on the cerebellar microcircuitry is largely known through lots of invasive studies and quite a bit of track tracing. But if we want to be able to do this in humans, or in live non-human primates or other species, then we have to be able to do this non-invasively. And the only method that we have available to do that is diffusion-weighted imaging. So I'm going to give you a little bit of an overview on diffusion-weighted imaging for tractography. So I will refer you to Henrik Lundell's talk for a much more advanced chat about diffusion imaging. So he presented just before me. So if you've seen it in order, you've already seen this. So we know that the brain is primarily made up of water, and we exploit the fact that the brain is made up of water to do diffusion imaging. And this image that I'm showing you here is obviously a big, beautiful brain. The only problem being is it's missing 50% of its neurons. So the cerebellum is missing from this and many other brain images and brain imaging studies across the last 30 years, 40 years. Okay, so we exploit this idea of the brain made up primarily of water. We look at the diffusion of that water across time with diffusion imaging, and then we exploit the properties of the ordered arrangement of axons within the white matter, and we're able to see greater diffusion along the tract, along oriented axons, and less diffusions or restricted diffusions across them. We take that and we compute local models based on it. So the most famous being, of course, the diffusion tensor imaging model. And DTI gives us things like fractional anisotropy, which I'm sure you've heard of and probably have used or may use. So that's the simplest model really to work with in diffusion imaging. There are other models such as constrained spherical deconvolution, which we're using exclusively. And that's, I think, all of the studies that I'm presenting here. Almost all this, yes, all of the studies are using constrained spherical deconvolution, which gives you a better distinguishability between angles of fibers that are crossing, for instance. So we then take those local models and we build maps of directionality, essentially. So here, this image down here shows you this is principal diffusion direction in all of these voxels. And to do tractography, we basically just set a region of interest where we track from, for example, right here. And then we follow how those principal diffusion directions are lined up. And for example, they could take us around the corpus callosum here and then terminate somewhere over here. So I won't get into the details on this, but basically you set a seed point and then you follow along the diffusion direction and compute where that streamline or where that tract would end, where that proxy for a tract would end. And most people now are using probabilistic tractography, and that basically just means that we end up with a probabilistic map of the connections between one region and another, where you have high confidence and lower confidence, depending on how high the probability is. Okay, we then take all of that, you do this probabilistic mapping, and then you map, for example, from the pons through to the cerebellar cortex. So here you can see the tractography from the pons directly through the cerebellar cortex and coming up from the ICP here in red and then out from the SCP in green in this particular example. So this is what it would look like with a simple tractography analysis within the cerebellum. So just to show you what this looks like, the cerebellar peduncles look like in dissection. So this, these two articles have a microdissection of the human cerebellum. So they're very interesting for understanding the arrangement of the peduncles in particular. And here again, you see the SCP coming up and going towards the thalamus, the ICP coming up from the brainstem, and the MCP coming out from the pons. Here's the ICP just by itself, and then the SCP up here. And then in this image on the right, this right here is the pons, and this will be the MCP, this huge fiber tract, or this huge fascicle coming through and terminating the cortex and the dentate nucleus. So just to give you an idea what this would look like in 3D, I'll take that same image we had before and overlay it. So then you can see where the pons would, the connectivity, type of connectivity you would see from the pons and how that would connect through to the cortex. Okay, so that has been looked at in a sort of bigger data way with a fair number of subjects by Van Barsten et al. In 2015. And here they did targeted tractography to identify each of the cerebellar peduncles. And here in yellow through to red, we have the SCP, so the superior cerebellar peduncle, which is coming up here, most easily seen on this slice. In green, we have the middle cerebellar peduncle going to and from the, or in this case, from the pons back towards the cortex of the cerebellum, and then the ICP coming up. So this is a probabilistic atlas that is now available in many software tools. I believe it's available for sure in FSL, and I believe also in SPM, or if not, it's really easy to get in. And you could use this, for instance, to do region of interest type analyses on the MCP, on the SCP, and on the ICP, if you were interested in that type of thing for example, a clinical application to look at differences between, for example, Alzheimer's disease and controls on these peduncles. But that's still pretty macro, right? That's still pretty macro. Those are really big sets of tracks, and that's not giving us specificity, right? I showed you at the beginning these sort of motor versus non-motor regions, and that's really the kind of the lowest, or sort of the simplest distinction that you can make between regions in the cerebellum and their functions, right? So motor regions and non-motor regions. But we know that the cerebellum is reciprocally connected to most of the cortex. And if that's so, then we should be able to parse out more information about its connectivity than just saying, oh, we have an MCP, oh, we have an SCP. We should be able to say within that region, what is connecting to M1, what is connecting through to the somatosensory region, et cetera, prefrontal cortex, et cetera. So that opened up a lot of new challenges, because we would really like to do that, get very, very detailed information about the connectivity within the cerebellum and between the cerebellum and the cortex. But that brings up a lot of challenges. And the primary ones for diffusion go around structure and resolution. So the cerebellum is quite small, even though it contains over 50% of the neurons in the brain, it's very, very well compressed. Or it's takes up a small amount of volume. And that means that resolution is key. So if you're trying to image something where you have very high packing density, then you need to have a higher resolution to be able to disentangle the effects within. The same can be said for angular resolution, so I won't talk too much about this. That's more of a diffusion-y thing, but because you have such a small region, you're likely to have lots of fibers that are interacting, so crossing each other, and your ability to resolve those crossings is low unless you have higher angular resolution. So you can't tell the difference. If you have low angular resolution, you may not be able to tell the difference between a track that goes like this and one that comes from somewhere close but is going in a different direction. So that also becomes very important when you look at intrinsic connectivity in the cerebellum. This last thing, which I already kind of mentioned here, is the density. So there's high density, which means that there's a lot of inputs and a lot of outputs to the neurons within the cerebellum. So the packing within the white matter is very, very tight, which then means you need a higher resolution to be able to resolve it more accurately. So this is an image from Flavio Dell'Acqua's paper in 2013, which was ex vivo imaging using diffusion-weighted imaging of the human cerebellum, showing the different scales that you can look at the data at and the amount of complexity that you can derive with higher resolution imaging. Another issue with cerebellar imaging is signal quality. So the cerebellum, of course, sits towards the back of the brain and inferiorly, just sort of behind the brainstem there, behind the pons. And because of that, it's low down on where the head coil sits, which means that you can have signal drop off. So coils have definitely gotten better, and I think Wietske has some good examples of high quality cerebellar collection in her talk, which is also in this section, so I should refer you to that. But the fact remains that when you lose some quality, you'll often lose a little bit of quality down in the cerebellum. So you have to be careful about your imaging down there. Another issue is something that I've seen come up quite a bit in the last 5 years, I think, and that's connectivity through to the cerebral cortex. So different labs have claimed that they're identifying connectivity from the cortex through down to the pons from the pons to the cerebellar cortex, and then from the cerebellar cortex back to the dentate nucleus, from the dentate nucleus back to the thalamus, from the thalamus up to the cortex of the cerebral cortex. And while it's possible that these studies have been able to identify this type of loop, it's a multisynaptic loop, and diffusion imaging is really not optimized for multisynaptic track tracing. So that is extremely problematic, and I think all of those studies have to be looked at with a very critical eye. Not to say that they're wrong, but just to pay attention to the details a little bit. Okay, so if we want to move beyond the peduncles, so beyond this macro view, what is it that we can do? And this is where I'm going to bring in some of the work that I've done with collaborators at Max Planck, and that one of my students is working on currently. And here we're going to talk about investigating the intrinsic connectivity of the cerebellum. So within the cerebellum, what does the connectivity look like? And we did this, so we even asked this question, can we do this? So this was a few years ago now when we collected the data. And here you can see, here's three different views of the brain, and this was our region of interest acquisition. And we acquired, within this very small region of interest, we required 0.8 millimeter diffusion-weighted imaging using the 7 Tesla scanner at the Max Planck in Leipzig, the old one, which has now been decommissioned. It also used dielectric pads. So we did find that with the head coil that we were using, we had quite a bit of signal drop-off, and we placed dielectric pads at the back of the head, which was relatively uncomfortable for some participants, but they did it, to try to recover or to attenuate the loss of signal in that region. And we were successful at doing that. So when we acquired our initial images, which is what you see here, this is one diffusion-weighted direction, one sagittal slice on top, slice on the bottom. They didn't look amazing. You can obviously tell some structure out of it, but they don't look super amazing. There's a lot of noise in there. But thankfully, in 2015, 2016, there started to be a number of individuals working on denoising algorithms for diffusion data, and that's now common and standard in all of the big packages. So MRTrix has a pretty good one, as does Pierre Coupe's group also has a very nice one. So the first thing that we did was to recapitulate what others had done and try to identify these cerebellar peduncles. And we did this in a slightly different way. We created these track density images, which are just super resolution images of the tracks, of the streamlines going between every region of within the cerebellum. And here the colors represent directions. So red is left, right, green is anterior, posterior, and blue is inferior, superior. So here we're looking through a cut plane of the cerebellum like this. And you can see the ICP coming up anterior to the dentate nucleus. The dentate nucleus is giving rise to the SCP, which is projecting medially and then superiorly. Comes to medially, goes anteriorly, and then it starts to go superiorly around here. And the MCP is coming out from the pons and going in through to the cortex and through the dentate nuclei. And then the other two images, you can see similar types of views. This is the MCP back here in yellow in the coronal view. And then in the sagittal view, you can see the dentate nucleus here, the ICP coming up, and the MCP coming around from the pontinuclei. Pons and the pontinuclei. Okay, so I looked back in the literature, and I was able to identify what I think is the first evidence for intrinsic cortical nuclear connectivity in the cerebellum, and this is all in non-human animals, primarily in cats. This work was done in, and here this is looking at connectivity between the cerebellar cortex, which is around on the outside here, and the dentate nucleus, or the deep cerebellar nuclei in most of these cases. So a combination of them. And the first study was from 1905, and then these really nice iconic images come from Jensen and Brodal's work quite a few years later, 35 years later, in 1940, and they have another paper in 1942. And in these, they did selective lesioning of cat cerebellar cortex, and then looked, so this is what they did here, they would lesion, for example, this region, and then they would look within the dentate nucleus or the deep cerebellar nuclei, and then map where degradation occurred. And they did this to try to recreate or recapitulate this map of how different regions of the cortex are projecting to the dentate nucleus, regions of the cerebellar cortex projecting to the dentate nucleus. This work was really picked up a long time later by the Strick lab, and here they worked in non-human primates instead of cats, and they were interested in identifying the complete loop that I showed you before between the cerebral cortex and the cerebellar cortex, identifying all of the waypoints along the way. And this is really beautiful work. I really encourage you to take a look at it. And what they created based on this was a summary map of the dentate nucleus, which shows the projections from the cerebellar cortex and the cerebral cortex. So how that entire loop goes through. If you're projecting from a motor region through down to a motor region of the cerebellar cortex, and then the motor region of the cerebellar cortex to the dentate nucleus, they would map that as motor. And that's what this map is. This is a summary map of the dentate nucleus on the right here, where this is anterior here and posterior here, dorsal, sort of an unfolded map. So it's, I'll show you a real image in a moment, or a closer to real image in a moment, but this is the unfolded map. So this whole region here, this large area here, is considered to be motor dentate. Region under here is considered to be non-motor dentate nucleus. And this is, just to clarify this, this isn't, this is only indicated as motor or non-motor based on the anatomical connectivity that they've identified with the cerebral cortex. So they use these viral tracers and then trace through multi-steps to identify where they go from the primary motor cortex to wherever in the cerebellum, and then they tag them. So this is really, really nice work. There's also been some functional work on this, also by Joern Diedrichsen's lab. There's the functional characterization of the dentate nucleus, and it maps pretty well to this work by Strick and colleagues. So that was great. That was in non-human primates, but there were, at the time, there was currently no evidence for corticonuclear projections in the human cerebellar hemisphere. So that's what we set out to do, to identify this intrinsic corticonuclear connectivity in the human cerebellum. And to do that, we used a tractography approach because we did it in vivo. And we identified different lobules of the cerebellum, and we classified them as either motor or non-motor. So in this case, lobules 4, 5, and 6 here, excuse me, which we classified as motor lobules, and CRUS1 and CRUS2, which we classified as non-motor lobules. And we simply took those lobules, we segmented them, and then we projected we computed tractography from then to the dentate nucleus. This is an identified 3D volume of the dentate nucleus. And then we looked at where those streamlines, where those tracks terminated on the dentate nucleus to try to categorize what the projections were from the cortex to the dentate nucleus. And when we did that, we were able to identify that these motor lobules projected primarily to the superior surface of the dentate nucleus, and the non-motor lobules primarily to the lateral and slightly posterior, or sorry, slightly inferior surface of the dentate nucleus. So what's interesting here is lobule 6 and CRUS1 have a fair amount of overlap here, particularly on the lateral portion. And that actually speaks to a point that I'll make towards the end, and that connects really well with the points that Joern Diedrichsen is making on how we should no longer be considering the lobules as the functional organization of the cerebellum, because this lobule, lobule 6 in particular, has approximately 50% motor and 50% non-motor type connectivity. So this is interesting that we were even able to pick it up here. So. Okay, so we took those projection results and then we did a classification or a segmentation technique on them, just a winner-takes-all segmentation. And we said, if there are more streamlines coming from lobule 4 projecting to this region, then we color it purple. For lobule 5, we color it blue. For lobule 6, we cover it red. And then the same for CRUS1 and CRUS2. And based on that, we came up with this final map of connectivity between the cerebellar cortex and the dentate nucleus. And that's what's shown here. And this maps very nicely to the work of Strick and colleagues. And so here, this lateral line that you can see here is approximately here on this map. So this is the superior surface, which is all this motor, and this is 4, 5, and 6, which are primarily motor. And that's what you see here in the anterior portion. We have 4, 5, and 6. We have unlabeled area here. And then in the more posterior version, areas back here, we see Cruce 1 and Cruce 2. And similarly, in the Strick work, we see these non-motor projections. So we're really happy with that. Very similar to the non-human primate work, but also indicated that the non-motor projections took up actually more of the segmented surface of the dentate nucleus than it did in non-human primates. So possibly we have this shift between humans and non-human primates and humans. Okay, so moving along and building on that work, what one of my students has been doing is investigating pontocerebellar connectivity. And this is, again, looking along the MCP and seeing where it terminates within the cortex. And here's some work that he's done just recently with 3T data. And here we've grouped lobules into motor, sort of putative motor. Here we've put 6 into the non-motor in this case, just to check how it goes. These are the images I had. And you can see the colors that you end up with are a mixing of colors. And at 3T, so this is where you can see the impact of resolution. At 3T, you have this quite mixing here in the terminations in the pons. Whereas we go to 7T, you see a little bit nicer segregation where you have these superior motor regions more inferiorly in the pons, and the same with the more inferior motor regions, and then the posterior non-motor regions projecting like more superiorly, and then the other colors are sort of a mix in between. And when you do the segmentation or classification on this, you can kind of pull out these motor regions here that are projecting, or that are projected to more, projecting from the pons in the more inferior portion. And then these non-motor regions, which are dominating of the pons. And this is consistent with some of the non-human primate literature. We can also segment out the MCP, so I won't spend too long on this, but you can do exactly the same thing and just sort of do slices through the middle cerebellar peduncle to see what happens to the tracks that are coming from the MCP, coming from the pons and terminating into the cortex, and label them according to where they terminate in the cortex. And here you can see that the superior motor starts sort of medially and going through the slices from left to right on this image, left to right here. You can see that spreads superiorly, and then the other ones have other types of shapes. So this could be really interesting information for people who are planning. I think could end up being very important for people who are planning surgery and such as well, because you know that if you come in this direction at this location, you're going to hit something that's more involved with non-motor, maybe language processing, than if you were in another location. So the final little piece I want to talk about here is looping the cerebral cortex in. So I know this talk is primarily, or should be primarily, about the cerebellum and connectivity in the cerebellum. But the cerebellum doesn't act in isolation, right? It's connected through to the cortex. So we do have some information from non-human primates. And again, Jeremy Schmamann and Tanja have done some beautiful work on this. And this is also track tracing in non-human primates, having projections coming from the cortex down to the pons and seeing where they terminate within the pontine nuclei, and then classifying according to where they came from in the cortex. And what you see here is a type of a very general arrangement that we see. So obviously we're not at the same level of detail as they were with diffusion imaging, because these nuclei are extremely small. But we see this general organization of non-motor projections going to the superior part of the pons, so up here, and then the motor regions are projecting more to the to the inferior part of the pons, so down here. So this green, blue connections similar to this. Okay, this is, to my knowledge, the only study that has looked at this in humans, compared humans and non-human primates with diffusion imaging. And this was work by Narendra Ramnani again. And here they didn't label the pons specifically, but they labeled the peduncles. So this is also very interesting work to take a look at. All right, so just to sum that up, I hope that you'll see here that cerebellar connectivity defines the function of different cerebellar regions, and that DWI can be used to infer cerebellar connectivity in vivo. I think that this has lots of potential clinical applications. So as I said previously, if you can do this type of classification where you segment out regions, that can be very easily used to compare different patient populations. And some of the techniques that Paul Noel is working with to try to segment out the MCP could be super useful for identifying, surgical approaches and that sort of thing. For example, for cerebellar mutism patients. So of course, as with every type of research that we do, there are lots of challenges that exist. And most of those challenges, I think, are down to diffusion imaging, but we're slowly trying to work through those. The last point I wanted to make is about moving beyond lobules, and I think I made this a little bit throughout, and you should really see this nicely in Joern Diedrichsen's talk. And here is just that I've, in my previous work, we've used cerebellar lobules as like a functional unit of organization, but we know that's not true based on the work of Randy Buckner back in, I believe it's 2011. And following with a lot of Joern Diedrichsen's work in his lab to show that the functional segregation of the cerebellum doesn't follow these lobular boundaries. So we have to start thinking about other ways to use connectivity metrics from diffusion connected to these types of maps instead of just lobular maps. Okay, with that, I would like to thank all of my collaborators and my students and you for your attendance, and please feel free to give me any feedback. Thank you.
Hello, my name is Henrik Lundell, and I'm a senior researcher at the Danish Research Center for Magnetic Resonance, which is in Hvidovre, outside Copenhagen in Denmark. In this talk of this cerebellum imaging session, I'm going to focus on how to image cerebellar subvoxel microstructure, mainly with diffusion-weighted techniques. So, in order, and as we will see in this session, in order to image the cerebellum, high-resolution imaging is really crucial in order to see the small morphologies that we are interested in. But the cerebellum is also interesting on many other length scales as well. So if we go to the microscopic length scale, we have some distinct features that we don't see elsewhere in the brain. And these features, cellular structures, cell morphologies, are at a much smaller length scale than we can image directly with the magnetic resonance imaging. So one of the favorite techniques for imaging cell morphology is to use diffusion-weighted MRI. I'm sure many of you know about this technique, but this is just a short cartoon showing the principle. If we look at isotropic free diffusion, that would be just a random terminal mode in the ocean, for instance. These will spread out evenly over time and similar in all directions. That's what we call isotropic. But if we have a water that is trapped inside white matter, in this case, the water will be limited in its ability to move by the boundaries posed by cellular membranes, myelin, etc. But diffusion will occur faster along axons as they are tubular, but not so much perpendicular to it. So the local diffusivity will in this case be anisotropic, and this reflects tissue density and also fiber direction, a crucial information, for instance, for tractography that we will hear more about in the next session. So a very common model a method to decompose a complex diffusion measurement is to fit the diffusion tensor, that is this ellipsoidal shape, and the size of this shape tells us a bit about how fast the diffusion is, and the shape of it, the anisotropy, tells us something about how the underlying microstructure looks like. Let's see here. So in this example, I have a conventional DVI dataset and a color-coded FA image. with a scan at 2.2 isotropic resolution, and I zoomed in on the cerebellum here. This is color coded, so if it's high anisotropy and fibers going left to right, it's red, and it's blue if it's fiber going up and down, and green if it's going anterior posterior. And you can maybe see that it's very hard to see the cerebellum here. I will try to outline it here. So this is the mid-sagittal section of the cerebellum. What you see here is the brainstem and the spinal cord going down in blue, and we have the pons with the middle cerebral peduncles going into the cerebellum. But obviously, this is not really a satisfying resolution to investigate the properties of the cerebellum. And many of the problems that are general for cerebellar imaging, like low SNR and low resolution, B1 and B0 inhomogeneities motions, are almost worse in the EPI acquisition that's usually used for DVI. There are different ways to correct for this and to make the acquisition more suitable for cerebellum. You can see Chris' beautiful examples of in vivo imaging in his talk. So one solution is to go to ultra high field where we get more signal, but we also have to work with the B0 inhomogeneities, which warps our images to a high degree. So all of these things has to be kept in mind when adapting diffusion weighted imaging for the cerebellum. But I'm also going to talk not so much about the acquisition optimization, but how we can provide better diffusion weighted imaging to get a more specific view on cerebellar microstructure. And here's an example of a post-mortem diffusion weighted imaging experiment performed on a vervet monkey, and this is at the resolution of 0.4 isotropic millimeter resolution. So the voxel volume here is almost 100 times smaller, or more than 100 times smaller than it is in this in vivo human data set. And this has been possible by the fact that we here used a preclinical scanner, and we could scan for days to collect high resolution data. So postmortem diffusion weighted imaging is a versatile and powerful tool when you want to perform method development and validation, as you can also do histology on your sample. You can also do collect very rich data set with a lot of parameters, which will guide you to find the sweet spots for investigating a particular microstructure in your in vivo settings. But the drawback is, of course, that there might be some differences between in vivo and ex vivo effects from fixation, for instance, of the tissue that alters the diffusivity. So this has to be kept in mind. So if we focus on this color-coded FA here, we can start to see that we can appreciate some of the finer structures of the cerebellum. When we look in arbor vita, we can see in green and blue the fibers going out to the more finer fissures of the cerebellum. We can overlay here the fiber directions. So here you can see that we start to get the information that we need to track out fibers or do tractography. But I said more of this in the next talk by Steele. So we start seeing white matter structures, but the gray matter is also quite interesting here. You can see that it's red and the fibers are pointing in plane here. So this is left to right in this sagittal plane. And what this is the outer layer of the cerebellar cortex. In general, gray matter in the cerebral cortex is not very anisotropic, but in the cerebellum it is. And what this is the molecular fibers or the molecular layer. In this layer, we have the very characteristic Purkinje cells lying in parasagittal planes, and those are interconnected by the parallel fibers, which runs along the fissures of the cerebellum. So they are really popping out here as an anisotropic structure in a conventional DTI analysis of the cerebellum at high resolution. Now, the conventional diffusion measurement and DTI or other measures derived from such data has some drawbacks that has to be either accounted for in the interpretation, or we will need some model assumptions in order to resolve different levels of complexity. So if we think of a very simple interpretation of the tissue, here are just some straight cylinders representing the axons. We can think of that this system will be well described by a diffusion tensor to some degree. But if we think of the real tissue, it's probably going to be more complex. We are going to have fibers crossing, bending, and maybe fibers of different constitution. So when we measure on a voxel level, we are going to measure the collective behavior of all those different microscopic environments. And the truth is that it's probably even more complicated, because we also have different cell types. So not only neurons, but we also have a large volume fraction of glia cells, astrocytes, oligodendrocytes. And in the case of a pathology or some kind of degeneration of the tissue, we have to take even more things into account. So what I'm going to describe today is how we can move beyond the traditional diffusion measurement to examine the microstructure of the cerebellum. And the key point here is to acquire the specificity about microstructure already in the data rather than in the data modeling afterwards. And I'm going to talk about 3 different approaches. One is to look at time-dependent diffusion, and I'm then going to move over to multidimensional diffusion, or one example of multidimensional diffusion applied to the cerebellum. And finally, I'm going to talk about a method called diffusion-weighted spectroscopy, which is a method where we are not looking at water, which is everywhere in tissue, but we are looking at the mobility or diffusion of metabolites that are intracellular and cell type specific. So I start with time dependence. So in free diffusion, the mean square displacement, or the mean displacement of molecules will follow this relation. It will follow the square root of time. So this is a quite characteristic shape for this kind of system. But if we have molecules trapped in some kind of enclosure, we are going to be trapped at some point. So in the beginning, we are going to measure the same thing as free diffusion before we start sensing the barriers. But as soon as we are going to starting to see reflections with the barriers, the displacement is going to be lower, and at some point we have completely explored the whole restriction that we are in, or the whole cell that we are in. Molecules can't move further. So in this case, if we look at this formula, the diffusion in restrictions is going to diverge more and more and more from this function. And this means that the diffusion coefficient here is the same as for free diffusion at very short diffusion times, but restriction makes the diffusion coefficient to attenuate or be lower with longer diffusion times. So this can be measured simply with the conventional pulsed gradient experiment that is used in different shades on different scanners. This can be the simplest way to do it is just to push our gradients very close together. But this will give us a very low p-value, a very low diffusion weighting. So the sensitivity will decrease rapidly when we go to those low diffusion time. So a trick to be able to measure short diffusion time and still have a high p-value is to use what's called oscillating gradient spinnacle, or OGSE. So you can think of the OGSE experiment as every wave here of the gradient is like one diffusion encoding, but then it's repeated many times, which will build up the p-value. in its most kind of simple interpretation. So this encoding can be characterized by an encoding frequency, and a high encoding frequency correspond to a short diffusion time. If we have two different restrictions here, one big restriction and one smaller restriction, we are going to measure low diffusivity at low frequencies or long diffusion times. But as we crank up the frequency, we are gradually going to experience free or go towards free diffusivity in the larger pores. So the diffusivity will increase in large pores, but for very small pores, it will be maintained. And also for free diffusion, like in CSF, the diffusivity is also not responding to this change in frequency. So this gives us a handle on a very specific feature of the diffusion signal. So we have performed measurements on the cerebellum with a type of OGSC, and what you can see here is the measurement at two different frequencies. And what you can see when we go to a higher frequency is that the change in diffusivity is not very high in a lot of the places like CSF or here in the brainstem, but you may appreciate that one layer of the cerebellar cortex is popping out, and this is the granular layer, the inner layer. So before we saw the high anisotropy in the molecular layer, but the granular layer consists of very densely packed neuronal cell bodies. And this is kind of the situation we see here. So when we go to the higher frequencies, the signal or the diffusivity in the cell bodies of the granular layer increases, and that's what we see here. And here's a Nissl stain, which is coloring the regions with high cell density of the monkey cerebellum, and you can see that this coincides with this inner layer of the cerebellar cortex. So going on to the next technique, that's multidimensional diffusion encoding. And what this technique is trying to resolve is different sources of anisotropy in a voxel. So we can think of the voxel as consisting of very small regions with a local diffusivity, and those local diffusivity might be the same as in the three first examples here, but directed in different directions. So the microscopic quality may be exactly the same, but the overall diffusion tensor that we'll measure as an average of these voxels will be very different. So if they are perfectly aligned, we are going to make sure the average is going to be the same as the microscopic environment. But as we add some dispersion of those small domains, we are moving towards an isotropic case. And in this case, the signal will be indistinguishable from the case where we have isotropic domain. So this could be just spherical cell bodies. So going back to the sequence diagram, so this is a conventional diffusion encoding where we have two blocks of gradients which encode and decode the position of spins. This means that we have marked all the spins during the period between those gradients. We have marked the position of spins in the phase of the signal. So this measures the displacement in a certain direction for every measurement. But we can also make a measurement where we measure, during the diffusion time, change the direction in which we measure diffusivity. So we sweep from, for instance, here 3 orthogonal directions. In this case, the measurement will be isotropic, which means that we are equally sensitive to displacements in all directions. And now we'll see how we can use this in order to disentangle the microscopic anisotropy. So here again, a directional encoding will capture all sources of multi-exponential decay or a variance in diffusivities. So if we, in this case, have a region here with isotropic diffusivities, which vary a bit, we are going to measure a multi-exponential decay, a sum of exponentials. And if we measure in one direction through this, which consists of the same anisotropic compartment, we are going to measure different projections of the same diffusion tensor, which is also multi-exponential. If we now add the isotropic diffusion encoding, we are, in the case of microscopic and anisotropic compartments, we are going to measure the same signal attenuation from each compartment, as the orientation of the compartment doesn't change the overall signal. So in this case, if they are all the same but differently oriented, we are going to measure a mono-exponential decay. And this difference here between those two signals is related to the underlying microscopic anisotropy, so how pointy those diffusion tensor are. So in this way, we can get independent information already in the raw data without applying any model or performing any assumptions regarding the microstructure. So these are just subtraction maps of the raw signal, and comparing the directional versus isotropic encoding, we get a measure of the microscopic anisotropy, and we can also get the information out about restriction size by changing or modulating the diffusion time in those sequences, similar to the measurements I showed before with OGSE. So if we zoom in on the cerebellum here, we can see that we have some quite interesting, that those two different measurement dimensions kind of classify different regions. So for instance, in white matter, we see a high anisotropy and some degree of restriction density. So the time dependence is apparent in the white matter and anisotropy as well. And this measurement is compared to an FA measurement not sensitive to crossing fibers. If we then look at the dentate and inferior olivary nuclei, so this is the dentate nucleus and here's the olivary nuclei, we can see that we have a low microscopic anisotropy and a low restriction density. So these are probably very entangled dendritic structures with maybe high permeability, which makes the anisotropy low, and we don't have any larger restrictions to sense with time dependence. And then most striking again is the granular layer, which has a low anisotropy, but a very high restriction density in this measurement, really boosting and lighting up here. And if we compare here the cerebellar cortex with other cortical regions, we can see a quite interesting gradient with a very high effect of time dependence in the cerebellar cortex, which is then dropping slightly to the visual cortex, and which is even lower than in parietal cortex and more frontal areas. And this is quite well known from histology. So if we look at the numbers of soma density in cerebellum, we have about 400 million somas per gram. This drops down to 150, so about 1/3 in the visual cortex, and it drops again by 1/3 to 50 millions per gram in parietal cortex and most of other cortical regions. And we can see that here, just the raw signal that we measure here on the y-axis is pretty well correlated to this soma density. Chantal Tax in Cardiff has done a kind of has used the same sequence in a kind of different approach, using the fact that the isotropic diffusion encoding will act differently on different cell types when we go to very high diffusion times. So if we have axons here represented by tubes, axons or dendrites, we are going to have a high diffusivity in some directions and low in some, but as the diffusion encoding is isotropic, we're going to always have a high attenuation in fibrostructures. Whereas we will have a maintained signal in small spherical cells where we have restrictions in all directions. And what you can see here in humans, when we crank up the b-value, we can see that the signal attenuates gradually, but the signal in the cerebellar cortex is really retained. So it seems that we are filtering out signals from dendritic structures and axons, but signals from spherical cell bodies like the cell bodies of Purkinje cells, and maybe even more from the granular layer where we have even more cells. This signal is retained, and that's what we see here at very high p-values. And here's another a coronal picture of the cerebellum where we can clearly see the cerebellar cortex popping out from the morphology of those cells. And this is here compared with a histological Nissl stain with inverted contrast. So you can see a very high cell density in the same regions here of the cerebellar cortex. So finally, I'm going to talk about the last derivative of advanced diffusion weighting techniques. And this is to look at metabolite diffusivity. So the main contribution to the MRI signal is water and maybe fat, which stands for a very high portion of the signal. But if we look at the FID and decompose the signal in different frequencies, we might measure some smaller peaks of the signal, which correspond to other molecules or metabolites. That's also that we can also find in the tissue. If we look at the difference in signal, here it's really, really high because the concentration of those molecules are very low. So it's about 10,000 difference. So it's about the difference between Mount Everest and your kitchen table. So those peaks here that you can see here in the bottom, here we have a zoomed in version of it. They correspond to different metabolites. And the interesting thing about those metabolites, or in the sense of for diffusion measurements, is that they are mainly intracellular, which means that we filter out a large contribution to the diffusion signal, the extracellular space, and we start getting into a more specific signal. And moreover, some of those metabolites are specific to specific cell types. So NAA is mainly a neuronal marker. So it's in axons, dendrites, and cell bodies. Whereas, for instance, choline is a glial marker and mainly in astrocytes. So we have handles on the intracellular spaces of specific cell types. So this kind of readout can be combined with diffusion weighting, and this is a collaboration with Itamar Runen at LUMC in Leiden. So here we add diffusion weighting to the magnetic resonance spectroscopy measurement, and we see an attenuation of the peaks, just as we see with diffusion weighting on water. But this attenuation is then related to the specific mobility of those metabolites. So the first in vivo DBS measurements were done already in the 90s, but has later on emerged to more different applications in, for instance, cancer and aging and MS, for instance. And this has been made possible by the advent of ultra-high field MRI, as this is a very low signal. So we have performed some experiments on some DDS or diffusion weighted spectroscopy measurements on the cerebellum at the 7 Tesla. And what you can see here is the spectra at different B values. So when we increase the B value, we see an attenuation in the signal, which is then specific to those different metabolites. So for instance, glutamate and NAA is mainly in neurons. So the interesting thing when we have this very specific signal is that we can make also some more specific hypothesis on how the signal will behave. So which was shown in gray and white matter by Christopher Kronicke in 2004, is that the signal follows in white and gray matter in the brain follows a very specific function, which is that of one-dimensional diffusion in different directions. So what we call sticks in diffusion. And a similar approach was recently done to water measurements by Jelferat. If we see a deviation of this, we can have some pretty strong strong guesses that this could relate to different microscopic environments. So if the curve is deviating, it's more flat, deviating downwards, we may have effect of those sticks actually being thicker cylinders. But if it goes the other way, we have a signal that is attenuating slower, and this could be an additional slow or dot isotropic compartment like that of a cell body. So in a paper we recently published, we showed again in the human that we have a very close resemblance to this function in white matter. And here's an example to that. Up to our measured point, we're very close to this stick function, maybe with a slight tendency below the line, which could be the result of a non-zero radius of the axons. But when we look at the vermis of the cerebellum, we see a quite different behavior. It actually, the curve goes the other way. And my interpretation of this is that we are starting to again have a handle on the cell bodies of in the granular layer. Now with a measurement which is even more specific to the intracellular spaces of those cell types. Still yet to be investigated is the astrocyte pool in this cerebellum, which will be possible by, for instance, looking at the choline diffusivity. So to conclude, I've been talking today about some different ways to enhance specificity in diffusion measurements and how this can be applied to the cerebellum. I have a few take-home messages here. So the advanced diffusion encoding techniques, where we change the traditional post-gradient spin echo experiment. If we change this to more advanced gradient waveforms, we are able to encode the time-dependent diffusion and isotropic diffusion encoding, which can provide us with information about microscopic anisotropy, which is not affected by the global dispersion within a voxel. And we can also get data which is extracting information about restrictions in the tissue. And these are extraordinarily high in the cerebellum due to the high soma density. And I also presented diffusion-weighted spectroscopy as a method and how it applies to the cerebellum, and also saw some results which clearly shows a very different microscopic environment compared to the rest of the brain. The data I showed was for the intraneuronal space, but in the future we are going to look more into astrocytic metabolites and differences in structure in cerebellum compared to other brain regions. And finally, I would like to add one point here, that those techniques are low resolution techniques, so we have quite large voxel sizes. We get a very specific signal from specific cell types, but this still has to be combined with information from high resolution structural data to know what the tissue composition is in those voxels. But luckily, this is the topic of upcoming talks in this symposium, so stay tuned. Finally, I would like to thank my collaborators and my funding sources. Thank you.